All right, everybody. So I'm just doing a re-recording of this presentation since we had some technical difficulties with the first one. And I'm just going to jump right into it and kind of skip some of these introductory slides about me. So my parent organization is the Minnesota Aquatic Invasive Species Research Center, or MASIRC, and we're interested in translational research, basically bridging the gap between understanding the biology of aquatic invasive species and bringing that research into the field where we can actually manage AIS. And my dissertational research is pretty in line with that. I'm researching common carp. This is the species I'm going to be talking about today. They have an interesting history with humans, being probably the most important aquaculture food source worldwide, and very important still in developing countries. So I have a little graph here just showing a couple of different common aquacultured species you might think of, with common carp being the most commonly the most abundantly produced worldwide, followed right by tilapia, and then some of the ones that we eat here more commonly, like catfish and rainbow trout, being a lot lower than these. So this just kind of puts that in perspective. Also, they can be sort of ridiculously valuable when it comes to the aquarium trade. So this is just a picture of one fish that sold for $1.7 million last year. But carp also have an interesting history with humans as being introduced very widely. In fact, they've been introduced into waters basically across all continents where temperatures are permissible. We can thank the U.S. Fish Commission for their introduction into the United States. And this is a map showing the distribution of where carp have been reported to CABI from 1966 to 2005. And only a few different countries have actually reported them as being invasive, which means they cause damage or impact human health. But they are introduced worldwide and have been shown to have negative impacts on shallow lake ecosystems. This is a picture, or this is a map of the USGS clustered specimen records, just as sort of a proxy for where carp are most densely packed in the United States and where they've spread to. They're pretty much ubiquitous in inland waters. And in Minnesota, it's estimated that 70% of lakes in the southern part of the state have reduced plant cover due to the benthic foraging activity of common carp. Koi herpes virus, or cyprinid herpes virus 3, is the virus I'm going to be talking about today. It was identified in 1996 and was first identified in the United States in 1998 in New York. Since then, it's been spread worldwide thanks to the international fish trade, and it's now an OIE reportable disease. It's also interesting as a potential biocontrol tool. There's been some interest in that, and that is my main interest in Koi herpes virus. It causes a pretty severe disease in aquaculture settings and has high mortality in aquaculture settings. And it also causes mass mortality events in wild populations as well, though wild fish are a lot more resistant to disease. And you can see a few of the lesions that are associated with it. This is a lesion on the gill. That's pretty common when there are severe infections. This is kind of the sunken mucous membranes in the head of this fish. It's called anophthalmia. And then also these external lesions like loss of scales and skin, kind of typical things you see in a lot of different infections. This is a map showing the distribution of Koi herpes virus in wild populations of carp in North America. So there have been reports in 17 US states and Canadian provinces since 1998. And this box is kind of outlining where our study area has been and the different places that we found Koi herpes virus and another virus called carp edema virus throughout the state and surrounding states. This box is also blown up in this map above it. And you can see that the kind of the distribution of where we found this virus in Minnesota. So we found a KHV in 13 different mass mortality events of wild common carp, and also 6 mass mortality events that were associated with mixed infections of KHV and CEV. So this work is a part of my dissertational research from 2017 to 2018 has been identifying a biocontrol agent, and we've focused more on KHV since then. So from 2019 to 2021, I'll be doing the rest of my work on that. And today I'll just be talking about a part of that, which is a description of the ecology of KHV in wild carp in Minnesota. So the purpose of this is to better understand the distribution and disease dynamics of what's a really important pathogen and is now becoming pretty common in Minnesota lakes, or is common in Minnesota lakes, and also to determine the potential value of KHV as a biocontrol agent for carp. And then finally, we're also hoping to obtain local isolates for lab-based research. So kind of the sampling and screening schematic that I'm showing here kind of gives an outline of what we've been doing for the last year in lakes where we already know that there's been a history of a mass mortality event associated with KHV. So going back into these lakes, we sample 180 carp. We did this in 5 lakes last year, but we sampled 180 carp and 150 native fishes, kind of split into 5 different species, but it's kind of a random grab bag of whatever we can find during the time that we have to sample. For carp, we take otoliths, which can give us a precise age if we section and look at the annuli in those otoliths. Right now, we're just working with length data, and so that's what I'll be talking about today, but we're hoping to get a more detailed data set with that. We also took whole blood and separated serum and sent off those samples for ELISA testing, which looks for anti-KHV antibodies or previous exposure to KHV. And that was only in a subset of our samples, so just 60 fish from 1 lake. We also take tissues and preserve them in RNA later to preserve nucleic acids. And that way we can actually go through and screen everything with qPCR, that's really sensitive test for KHV DNA. And samples that are positive from either natives or carp, we can then test with an mRNA detection assay that's specific for reproducing virus. So that way we can actually test whether the virus is replicating or not. And then finally, we take an aliquot of our tissues and submit them to virus isolation as quickly as possible to see if we can get some isolates out of all these samples that we're taking. So this is a summary of the information from 2017 to 2019, just kind of a spatial look at things and kind of a tabulation of the numbers of places that we found these viruses. So the first map in green is showing mortality events of carp and of native fishes, or mixed mortality events, and also surveys of populations of carp with no known previous mass mortality associated with KHV or anything else. And in black, the black dots were showing the carp mortality events. There's 29 of those. Mixed mortality events with native fishes, there's 14, and those are in the pink. And then the empty circles are showing the healthy carp populations. We've surveyed 18 healthy carp populations. This map in blue is showing the carp-only mortality events and kind of tabulating those based on the pathogen that we found. So we found KHV in 13 of these, carpedema virus in 5, and then also mixed infections in 6 other lakes where there's basically both viruses were present. We also looked for spring viremia of carp virus, SVCV, and we did not find that anywhere. Only 5 of the mortality events that we've investigated have been negative for any of these viruses. And then finally, the surveys of healthy carp. So this includes the 18 populations in red that had no known previous history of KHV. And then there are 7 lakes here that we've actually gone back to a lake that we knew had a previous history of KHV and looked for the virus again. And we found KHV alone in only 2 of those, but we found both like sort of mixed infections of KHV and CEV in 5 of them. KHV in Minnesota, we've done a little bit of genotyping, and we've so far determined it's of the European lineage, so KHV European rather than Asian. Based on a recent study looking at the sort of the thymidine kinase gene, the TK gene, it has a few useful polymorphisms for differentiating between Asian and European genotypes. these authors found that the Asian genotype seems to be arising in North America more recently than the European. So that might be a clue as to the etiology of our virus. So from 2017 to 2019, this is a little bit more information about these mortality events. It looks like we had a lot of cases that were reported in 2017 and 2018, and most of those were KHV. For the vast majority of them, we were just KHV alone or KHV in association with CV. And then in 2019, there were fewer mortality events reported overall, and almost all of them were CV alone or in co-infection with KHV. And then in 2020, we've had 6 mortality events reported so far, which is pretty early in the season. So there may be a lot of them, but we don't know what those are yet. Also, the temperature and seasonal window for KHV mortality events appears to be between 20 and 24.5 degrees Celsius, and all of them occurred between June and September, and lasted from 16 to 23 days in duration. We also did some rough mortality estimates at these events, not always done by professionals, but basically we were asked whoever was visiting the site to estimate the mortality from 1 to 50, 50 to 100, 100 to 1,000, and 1,000 to 10,000. And most estimates were 100 to 1,000 CARP. There were some that were up to 10,000 and some that were much lower. And CARP sizes ranged between 23 to 71 centimeters. So that's a pretty wide range. There could be a lot of different ages in there. But generally, almost everything in that range would be sexually mature. The majority were between 30 and 40 centimeters in range. So survey site results from lakes where we knew there was already a history of KHV mortality, and we went back and looked again. We found a low prevalence of new infections. So that's a low prevalence of samples that were positive for qPCR tests in lakes with historic outbreaks. So 3 lakes had less than 2% of samples being positive, and 2 lakes had 12 and 23% of samples being positive. Lake Elysian, in particular, had 23% of samples being positive, and there was also evidence of continued transmission and mortality, since we found dead fish on that lake as well that all tested positive for KHV. So this inset here is showing the location of Waseca County in Minnesota. There was a lot of events clustered here from 2017 to 2019. Just kind of a blow up of that location with the lakes that tested positive for KHV in 2017 and 2018 are marked in red. Gorman and Sabre are 2 of the lakes that were on that low end of prevalence, so very few samples testing positive. And then Elysian down here in the south is right there. And the locations where we found dead fish are marked in red on this map. We also found evidence of acute disease in 15% of individuals in this lake from what we sampled. And the mean qPCR copy number was 253,000 copies per milliliter of tissue, which based on our testing is a pretty high viral load. We were also chose to do the ELISA and qPCR testing subset in this lake, which turned out to be really useful. When we pair these 2 tests, we can combine the ELISA with PCR. If they're both positive, then we say this is a persistent infection or an individual who is convalescent and is still shedding the virus. And then ELISA positive and qPCR negative would be a latent infection. So they have recovered and they're no longer shedding the virus at that time. And then qPCR positive, but ELISA negative would be an acute infection. And there was 1 or 2 cases where there was pretty severe disease signs. And we assumed that those were individuals that were ELISA positive, but we assumed that they were still new infections. So looking at the prevalence of these disease states in this lake, acute and persistent infections were actually the most common. So that's kind of like an outbreak type scenario. Those were the most common disease states that we found. This is the second graph is showing the relationship between carp length and disease state. So sort of like between age and disease state. What's the association there? And basically what you can see here is that if you're a smaller fish, you're more likely to get an acute infection or die from one, which makes sense since a lot of these individuals are sexually mature, but they're probably only participating in spawning for the first or second year of their life. And thus maybe wouldn't have had any previous exposure and would be more susceptible. There's also a relationship between the ELISA value, so the titer of antibodies, and the fish age. So it seems as fish age, they have a higher and higher ELISA value. So maybe they're mounting a better and better immune response as they get older. The colors in this plot are just showing the different disease states, so you can see that along with the data. The final interesting thing about this population was that there was an association with of disease state with sex. So we found that there were more acute infections and deaths among males than among females. And Elysian, based on our sampling, is female dominated, with 67% of the individuals we sampled being female. And this is not typical for wild carp. You'd expect a 50-50 ratio. And the potential mechanism here is that males do mature faster than females and respond to sex hormones sooner or earlier in life than females do. In addition to that, spawning females will lay all their eggs and basically only spawn one time, while males will participate in spawning with multiple females throughout the entire spawning season. So there's more opportunities for close contact during permissible temperatures. So finally, we were also hoping to isolate a local strain of KHV. So the approach here was kind of twofold. One was to basically just take tissues from all the fish that we were sampling and take aliquots of them, get them to the lab and subject them to cell culture within 48 hours. And then the other approach was to take a live subset of these fish and bring them to our containment lab and do sort of like non-lethal screening for KHV. So we can just take a gill swab and test them to see which ones were positive. And if we get positive fish, we would leave them in the tank and thermally stress them by shifting the temperature up and down to try and increase the shedding of KHV. And then we could use their secretions or tissues or even just cohabitation to transfer the virus to naive lab-raised juvenile carp and koi in order to start new acute infections that would be much more likely to produce an isolate on cell culture. And we found that we had a really high success using that in vivo transmission model, but very low success of isolation directly from wild carp tissues. So just to kind of show what we did here, we tried to just take tissue pools. We had 116 of those tissue pools, and none of them produced an isolate. So none of them formed cytopathic effect on cell culture. While tissue pools from temperature stress wild carp, we tried this one time and it did work. We were able to obtain isolate that way. And also we were able to obtain isolate 7 out of 7 times that we tried the in vivo infection model. So the conclusions from this presentation that I want to show is KHV is widespread in wild carp populations in Minnesota, but may not be continuously shed or detectable during permissible temperatures. Also, KHV and CEV is a common cause of most spring and autumn die-offs of carp in Minnesota, but it's unlikely to be associated with mortalities of native fishes. So it was just interesting that we did most of the mortality events that were reported to us and that we investigated were associated with either one of these viruses, but none of the mortality events of native fishes or mixed mortality events with native fish and carp did we detect any of these viruses. Also, populations with histories of KHV may have ongoing mortality in subsequent years, and it may be associated with young sexually mature fish, especially males. And that wasn't true in all of the lakes that we looked at, but it was true in Elysian. Additionally, anti-KHV antibody titers are higher in older fish. So that may mean that there's going to be higher resistance to KHV morbidity in fish that have reactivated infections over time. And also they may be less likely to shed the virus over time. The in vivo infection model finding was a more effective method of obtaining study isolates from wild populations than using tissue, even if we're trying to rush it or putting it in a special medium to make it to prepare it for cell culture. So the next steps for me are to continue to investigate new and local outbreaks of KHV, hopefully refining our sampling and mortality estimation, and also to obtain a few more strains of KHV. I'm also hoping to evaluate aspects of the pathogenicity of KHV in lab trials. So I've gotten to do this a little bit already, and didn't have time to present some of that data yet, but hoping to do more of that. Also, in the lab and in the field, we're also checking to see what the host range of KHV is using native fishes in lab-based trials, and also screening these populations with previous histories of outbreaks to see whether KHV is carried in any of those native fishes. And then this summer, I'm doing a more in-depth cross-sectional sampling throughout the season on Lake Elysian with the qPCR and ELISA testing strategy to get a more in-depth view of the ecology of KHV in that lake. I'm also hoping to develop collaboration for development of a disease or population model, which could be used to sort of test some different theories about how KHV could be used, along with other management tools and a biocontrol strategy. And then the synthesis of all this would be an evaluation of KHV as a biocontrol agent for CARP, with as much information as possible on the ecology and disease dynamics. So I'd like to thank everyone who's involved in this project. There's a lot of people who have helped out with this, a lot of students and technicians, and collaborations with the DNR and other companies. And also our funding with the Environmental Trust Fund and Minnesota Aquatic Invasive Species Research Center. All right, thank you for your time once again. And here's my contact info if you'd like to reach out.
Awesome. Thank you very much for re-recording that, Isaiah. I was able to capture the last little bit with the questions.
Okay, great.
So I'll try to merge these two into one recording.
All right. Thank you.
All right. Thank you, Isaiah. We have time for just a few questions, probably. We've got one here in the chat from David Scarf. He compliments you on the presentation, by the way, and asks, given that all herpes viruses will reside in the neurons for the life of the host, what's your opinion about using KHV to control populations of wild carp?
So, I mean, I've been trying to wrap my head around this. I'm not sure about this, but I think the jury is still a little bit out on whether KHV like actually establishes latency. I know that there is some detection in nerve tissue, and there's also detection in B cells, B lymphocytes. So we're not really sure exactly how that works, but it does seem like the virus sticks around over time. And I think that that could actually be a benefit if you're thinking about this more in an augmentative approach, rather than thinking about sort of a classic biocontrol approach where you would introduce an exotic pathogen and hope to sort of like have like the shock to the system type of, kind of decreasing a population all at once. I sort of wonder whether there's sort of these more long-term effects, such as sex ratio biasing, that could be used in association with another biocontrol tool or other management technique.
Okay, we have time for one more question. If somebody has one, they could either unmute themselves and ask it, or put it in the chat box.
Hey Nick, I was writing one, but I'm pretty slow typer. Isaiah, this is Esteban from UC Davis. Great.
Oh, hey, how's it going?
Good, man. Isaiah, I got a question for you, and I came a little bit late, to the chat, so I maybe missed it. Actually, it's two questions. The first one is, how are you doing your challenge? if it is by injection or by maceration of tissues, from dead ones, or cohabitation. How is it that you're doing your challenges, from the infected animals into your challenge ones?
So I did a few different ways. I did take tissues and homogenize them, treat them with antibiotics, and filtered with a 0.4 micrometer filter, and then injected those by IP injection. But one of the ways that I found just to be the most effective was just to take a mucus sample from infected fish by swabbing, and just to spread that along the outer surface of the recipient, and that worked really well.
Okay, excellent. So yeah, so that will be a great way to do it. And my other question and kind of a comment as well, it's very interesting. You're finding, with the titers, and the correlation, with the age of the females, of the adult females. And I guess, my comment follow up, from Dave's question is that I actually think that really indicates, that the potential of using KHV as a biocontrol will fail, because those females that could potentially be, reproductively active, from here on, the fact that they have positive titers and that they have the higher titers actually could indicate, that they were able to develop protective immunity. And as long as they don't die from KHV or any other thing, that they will continually to be reproductively active, and could potentially continue the transmission of KHV, and they wouldn't be, every time that you challenge them, you will basically be giving them almost a boost for the virus, and you will just keep, getting a feedback, to generate, more antibodies, and again, those animals that are as you are mentioning, at adult reproductively active females, wouldn't be affected by the virus.
True. Yeah, and I think that's what makes this not I don't think it should be viewed as a silver bullet strategy. It's not the type of pathogen that you're going to be able to introduce into a population and expect the population to even be reduced completely over time. There's no reservoirs that we know of for the virus, and the mortality isn't that high, and there's also long-term immunity. So I think that however KHV is used, it would have to be used in an augmentative approach. So you'd have to be thinking about how you're going to remove those long-lived individuals, and hoping that KHV is going to have more of an impact on those younger individuals that are recruiting into the population, because those are actually harder to control. And there's not a lot of methods for controlling recruitment, while there are lots of methods, mechanical methods, and just kind of normal methods for removing adults from populations.
Yeah, that's a great one. Yeah, no, I completely agree. And my last question, is from the isolates that you have you by any chance genotype the ones that you have been able to recover, either by just PCR or even better the isolation? And I'm wondering if you have challenged them, because we have a number of KHV, particularly from pet koi. And I think they're different, from the ones in wild, if I recall. But I know that there is at least 2 different types in the U.S. of KHV, and I'm wondering, if which genotypes do you guys have in Minnesota?
Yeah, so it looks like everything is European genotype, and they're really similar. I don't know if you're familiar with the Polish POB3 strains, but they're really similar to those. So they have, some of those indels that are sort of like the Asian lineage, but overall they're most similar to the European lineage, and they cluster with European isolates.
So sorry to interrupt, Isaiah. We're gonna have
go ahead. Sure.
We're gonna have to keep on moving.
Come on,
European.
Nate.
Yeah,
European. That's what they are.
We have nearly a full genome sequence and VNTR analyses in progress right now, so we can follow up more.
Yeah,
on
I would
that
love you to do some challenges, with the Asian one also. If you guys don't have Asian, we can send you guys, and you can do comparative. That would be awesome. Thanks for the offer.
That would be really cool. Yeah.
All right, Isaiah, let me know, and I will send you guys the isolates.
All right, thank you. We'll be in touch.
All right. Thank you very much, Isaiah. It's a bit anticlimactic without the applause, but I'm sure we're all doing that in our living rooms right now. So we'll shift gears here, and sorry, Brad.